Hi, everybody. It's a great pleasure today to have in the HIAAD series of seminars Christos Gkogkas, a research director in the Biomedical Research Institute of the Foundation of Research and Technology at Ioannina. Christos is an expert in neurobiology, and the talk today is going to be on regulation of protein synthesis in memory and neurodegeneration. Christos, thank you very much for accepting the invitation of HIAAD to give a seminar in this series, and the floor is yours, and we are very happy to have you with us. Thank you.
Thank you very much for the kind invitation. It was really nice to have the opportunity to present here. It's something I'm going to talk today about two things I don't often talk about. So, and actually putting together on the same presentation is actually fitting this audience. So I put it together, having in mind the audience and the initiative, which is actually a great initiative for Greece and for the world. So I'd be happy to learn more after I'm giving my talk today. Maybe we can discuss more about other things that concern HIAAD. So let me see if my slides are moving. Please feel free to interrupt if you have a burning question or if I'm over my time. I'm happy to stop at any point. I always start my talks by talking about the people that do the work. And I've been lucky over the years when I started as a PI at the University of Edinburgh and now at Forth to work with very talented individuals. I don't get to name them one by one, but today the two projects I'm going to talk to you about are the PhD work of two talented PhD students at Forth, which is the second project we're going to talk about today. And then the first part, which has to do with memory, is the PhD work of Constance in Edinburgh and a little bit the work of Stella. So there are going to be three parts. I'll give you an introduction to the subject matter and the science that we do in my lab. We're going to talk about regulation of protein synthesis and memory, and then again, regulation of protein synthesis and neurodegeneration. And speaking about proteins, I'm a biologist by training. That's my first degree. Then I had a spell with bioinformatics and neuroinformatics for a few years, only to come back to neuroscience. And throughout the years, I've always had a fascination about proteins, and any biology textbook will tell you that proteins are the molecules on which life depends. Of course, it's a protein-centric view of life, but they do come in different shapes and sizes. And really, the questions we're asking in my lab is which proteins are important for life and for its manifestations? Where are they in the cell, in the body, in the different tissues? And by answering these two questions, you can ultimately answer why that is. And we try to do that in a way that we look at the transition from healthy to disease states. So I'll be talking about that today some more. And my involvement with proteins during my postdoc started with my interest in how proteins are being made, so how protein abundance is dictated in cells. And there's a key concept in the field. Sorry, I have to move a little bit the Zoom windows because I really can't see my slides. If you can give me 1 second, sorry. Yeah, here it is. So the concept of translational control is the sum of regulatory events which dictate how much protein is synthesized per mRNA molecule in the unit of time. And this is a mathematically incorrect formula, but it gives you an idea of how a cell could boost protein abundance by intervening at this step of translational control. And within the gene expression pathway that we all know in biology, obviously for the past many years, the focus has been on transcription, and indeed, transcriptional programs are critical for dictating protein abundance. But it's been clearly shown, for example, in this key study in 2011, that in some cells, in many cells actually, the step of mRNA translation and all the events that I just described you with this term translational control, they can dictate up to 70% of protein abundance. So by measuring translation and protein synthesis, we could predict protein abundance better than we do with transcription in certain cases. That's not always the case. And of course, mRNAs and proteins are molecules, so they do get metabolized, they get degraded, and all that contributes to protein abundance. So protein synthesis has three steps, which are very similar to the three fates and the thread of life. So there's initiation, there's elongation of protein polypeptide chains, and then there's termination. And initiation is the rate-limiting step of protein synthesis. That's where I've focused most of the work I've done initially as a postdoc and now as an independent PI. And translational control and initiation is actually the sum of events that happen when an mRNA at the 5' end is recognized by this complex of initiation factors, about 20 of them. A key one that, if you need to remember anyone, is this protein called the IF4E. It's the cap-binding protein, and it's believed to bring everything together. Then there are scaffolding proteins like 4G. And all this complex does is it binds to the small ribosomal subunit, and it travels down the 5' UTR. It unwinds it, so it uses energy in the form of ATP and helicases like 4A and 4B to unwind the secondary structure and deliver the small ribosomal subunit at the initiation codon, where it will recognize the 60S ribosome. Together will give the ATS, and elongation will happen. So initiation is all the events that happen here just before the start codon. And why would nature deliver such an elaborate mechanism for doing this thing? Because it offers a lot of regulation for controlling life, I would say. And actually the structure of 5' UTRs is critical. It dictates in many cases based on RNA binding proteins that might bind, or the speed of initiation or the rate of initiation. It dictates translational efficiency, which is how effectively protein is going to be produced to a large extent. The formation of the complex that I just described to you in a very simplistic way. And the work that we and others have done is revealing this subset of mRNAs, which I would term 4E sensitive. It means that they require EIF4E for their translation. It could be a paradox where global translation, so overall translation of proteins, is going down, but these 4E sensitive mRNAs are going up in different scenarios. And one family of proteins that does that is this family of inhibitory proteins called 4EBPs, one of them in the brain, that we're going to talk about today, is called 4EBP2. And it competes with EIF4E for binding to the capsids at the very first step of initiation, where this control takes place. And while that is a paradox, viruses have been doing that for billions of years. They've been hijacking the host's machinery to preferentially translate. So viruses that have mRNAs, RNA viruses, they really depend at the very first step on 4E dependent translation, and that's why 4E is targeted in many types of viral infections. Actually, in COVID now, there are several startup companies that started specifically targeting EIF4E to inhibit the viral replication. So viruses are taking advantage of that machinery, and their mRNAs have structured 5' UTRs, which are preferentially recognized by EIF4E. And that's how they've survived for billions of years on Earth. And actually, an experiment that was done in the lab where I did my postdoc, they did show that when you knock out these 4EBP proteins that regulate translation, that render cells completely resistant to a viral infection from this vesicular stomatitis virus, VSV. So you can see either BP1 or BP2 deleted. There's almost no viral infection. And apart from the biophysical and molecular initiation, the mechanisms of initiation, because protein synthesis is a metabolic process within the cell, it is controlled by signaling pathways which help the cell sense the extracellular environment and convey that message all the way down to initiation of protein synthesis and either switch it on or switch it off. And two big pathways which you might be familiar with is the PI3 kinase AKT, TSC mTOR pathway, which actually regulates also general translation, but it has two main endpoints on initiation, one of them being the phosphorylation of 4E binding proteins, which when they are phosphorylated, they cannot longer bind to 4E, and 4E is free to initiate translation, but in their hypophosphorylated state, they're repressive for translation. And on the other hand, it can also phosphorylate the kinases like S6 kinases, which directly phosphorylate ribosomal protein S6 on the 4TS ribosomal subunit. And on the other hand, apart from this pathway, there's the MAP kinase pathway, which starts from RAS all the way down to ERK, and some other kinases such as MINC, which directly phosphorylate the IF4E to initiate translation. And this is by no means a comprehensive view of signaling. There are other signaling pathways which might impinge upon those, but it gives you an idea that apart from the sheer regulation from the 20 or so initiation factors, there's also signaling, which is extremely important for this process. And to answer the where question, which I told you initially, I think neuronal cells are a very good model because the all the regulatory variables that I described to you. They take an additional dimension here, which is location. And neurons are compartmentalized cells, so protein synthesis does not only happen in the cell body, but also it can happen in the axons or growth cones during development and presynaptic terminals, or in more mature neurons in their dendritic tree, on dendritic shafts, on spines and postsynaptic terminals. And really, in these cells that are highly compartmentalized and their processes are far away from the nuclear control, it's really important for the fidelity of processes such as translation to be maintained for synapses, because that ensures the information flow from the presynaptic terminus to the postsynaptic terminus. And this is what underlies all complex functions of the brain. So it's really a key process to maintain in the brain. And to prove why that might be important, actually, we knew that from the 1960s and the work of Agronov, who showed that goldfish memory actually depends on protein synthesis. They injected high concentrations of an antibiotic called puramycin, which inhibits protein synthesis by binding onto the ribosome. And he did show that learning and memory was impaired. The animals could no longer remember a task that was based on inhibition. And while this is the case, and this is knowledge from many years ago, we tend to remember this just by the fact that goldfish have a short memory of minutes, which is wrong, because you can see the experiment they ran with memory was on several days and several hours. So that myth is not exist. And coming to today, from the 1960s, memory, either in the form of sensory memory or short-term memory, which is something that lasts up to a few hours, or long-term memory, which lasts up to from 24 hours and onwards, work from many labs that has also led to a Nobel Prize for Eric Kandel has shown that new protein synthesis is required for the maintenance of long-term memory. But what has been elusive in the field is how do the neuronal circuits which underlie long-term memory establish themselves? Which are the genes that underlie memory? Which are the memory genes? And being a neuroscience lab starting in Edinburgh in 2013, we had a discussion with our students that we were using a lot of behavioral analyses for a lot of our mouse projects. And one of the tasks that we used routinely was this contextual fear conditioning, which is a form of learning in rodents. And basically, it's coupling an environment, let's say a box where the animal is, with a mild foot shock, which is called an unconditioned stimulus. And then the animal learns to associate these two stimuli, and then elicits a behavioral response, which is part of the fight, flight, or freeze response that animals have in nature. And actually, in that context, freezing is the predominant behavior that animals show. And it's been shown by many labs, like the Ledoux lab, that if you do lesions or pharmacological inhibition of an area of the brain called hippocampus, and in particular the dorsal hippocampus, that this behavior is highly relies on this brain region. And the psychological underlying concept is that same as the Pavlovian, classical Pavlovian learning. When an animal such as a dog is presented with food, they show this unconditioned response, which is salivation. A neutral stimulus like a bell does not elicit the same response, but when the food and the bell are presented together, then the animal will salivate by only having the presentation of the bell. And this is the same principle on which the contextual fear conditioning test is based on. So when we started doing this, we set up our experiments and our controls, and we realized that when behavior is measured in this text, at the time, almost 90% of contextual fear conditioned experiments, they didn't have a control that we thought to be important, and actually we do show in this study that it is. So if you remember, to elicit long-term memory, you need to pair the context to the mild foot shock. And actually the protocol is the animal gets into the box, it receives 2 mild foot shocks, which are 30 seconds apart, and then it's removed from the box. 24 hours later, it's put back into the box, and it displays a freezing behavior of about 60 to 80%. But if the animal is, if it's home cage, it displays a very small amount of freezing behavior within the same time. And if it's exposed to a same duration shock, so 2 and 2 seconds of the same amplitude, but removed quickly from the box, then it does not associate the box with the stimulus. So 24 hours later, it does not remember the box that it's been in. So long-term memory has not been formed. So the question for us was, it's a clear distinction, and this is a clear control for behavioral paradigms. Like there's no behavior here, right? So you could admit that it's the same as the home cage, but is that the same in terms of gene expression, which is required to establish the neural circuits that underlie these behaviors? And for the point of transcription, for many years, there are technologies such as DNA microarrays and RNA-seq. You could look that easily. I was very lucky in 2011 to visit the lab of Jonathan Weissman at UCSF, who at the time was developing a methodology called ribosome profiling. And what they do in ribosome profiling is they measure translation genome-wide. And this is a methodology called translatomics, which was missing at the time from the field of omics. So what we did is we decided on an arbitrary time point of 20 minutes because we thought that would give enough time for protein to be synthesized in our model. And we trained the animals in the 3 groups that I showed you earlier and extracted the hippocampus and then dissected just the dorsal hippocampus, the area that I told you was shown to be associated to CFC. Then we isolated mRNAs associated with ribosomes, and we did 2 preparations. One where we digested all RNAs with a nuclease, and only the part of the mRNA which is actively translated by the ribosome was protected here, and this is called a footprint. We then transcribed those into a library and subjected it to RNA-seq, and we got to measure a proxy of where the ribosomes are sitting on all mRNAs in the genome and how many ribosomes are associated per mRNA, which is a proxy for translation. At the same time, by measuring transcription for total mRNA, we got a measurement for transcription, in which the fraction of the 2 gives us the translational efficiency, which is what we defined earlier as the potential of mRNAs to give protein. And when we looked at the data, 20 minutes in the hippocampus for the 3 groups, I think the first striking result was that there were pervasive changes in gene expression in the shock group, which was shocking, but pun intended. And when we looked at the other groups, for example, the contextual free conditioning group, so genes going up or going down, we did see actually that, yes, the neurons and the dorsal hippocampus, they do elicit a gene expression program there. But looking carefully at the genes, we saw a significant overlap both at the level of translation and at the level of transcription between the different groups. And looking more carefully at the genes which were upregulated in the shock group and the CFC group, for translation, there was a small overlap, but for transcription, we have a big list of genes. And what is striking is that it's 99% occupied by immediate early genes, so genes that are activated when neurons are activated by neuronal activity. So why is that important? A huge fraction of today's neuroscience is based on correlating behavior with in vivo imaging that is based on reporters which are fused to fluorescent proteins such as GFP, and they have promoters which are activated by genes such as CFOS. And our study was one of the first to show that the activation through these promoters, which we see in the immediate early genes, might not be as relevant to behavior as people think it is. When we looked at the gene ontology into the groups that we had identified in this experiment with the shock-only control, we saw something that was of interest to us in terms of translational control. And that is that in the genes that were differentially translated with ribosome profiling, we saw a clear signature of the initiation factor 2 alpha pathway, for which I haven't told you anything. And in contrast, the categories were very different in the shock-only group. It was mainly some stress pathways, but also some mitochondrial and oxidative phosphorylation genes were also part of this data set. And one of the 20 initiation factors is EIF2. EIF2 is actually very important, but it runs an initiation process which is parallel to the 4F complex that I described to you earlier. It's called the 43S pre-initiation complex, which includes EIF2 fused to GTP, the methionine, tRNA, and the 40S ribosome. So all these things together, they make up the ternary complex. And when this is formed together with 4F, it gives us the 48S initiation complex. Why is EIF2 important? Is because its phosphorylation status dictates global translation to a large extent. So when EIF or EIF2 is phosphorylated, and that is when some of its kinases shown up here or its phosphatases work in that way, either dephosphorylated or phosphorylated, then global translation is down. And what happens, though, paradoxically, is while global translation is down, the translation of certain mRNAs, which contain what are called upstream open reading frames, such as ATF4, is up. ATF4 is a known repressor of CREB, which is a transcriptional activator that regulates learning and memory. And that was a huge leap in the field when this link was made between EF2 alpha, ATF4, and CREB. And almost 20 slides into the presentation, it's my first mention to Alzheimer's disease and other neurodegenerative disorders by saying that apart from translation, EAF2 alpha is also linked in a pathway that is upwards of translation. It's more proteostatically related. It's called the integrated stress response. And it's been now shown convincingly, both in animal models, but also in humans, that the ISR is activated in several disorders such as Alzheimer's and Parkinson's disease. While conversely, we and others have shown that in neurodevelopmental disorders such as Fragile X syndrome, the phosphorylation of EF2 alpha is low, so the ISR is low. So we went on to confirm this by looking at the messages which were found in our ribosome profiling using bioinformatics to analyze the sequence of the 5' UTR. We basically looked at the GC content, the energy that is required to break these structures, which is the energy that the helicases would have to employ to resolve the structure, and then the length of the UTRs. And not many features stood out, even though we saw shorter 3' UTRs and actually higher energy, higher Gibbs energy for the upregulated translated genes. The main feature that stood out, comparing it to the shock group, was that mRNAs that contain upstream open reading frames were clearly represented and enriched in this group, suggesting that the gene ontology category we found is reflected in the genes that we see to be differentially translated. And actually, these are the genes which are not affected by shock. They're only affected when the context is paired to the shock. So they are more related to the behavior. We did not see any changes in global translation, so we repeated the experiment and then isolated again the dorsal hippocampus, and this time used a sucrose gradient, which separates ribosomes based on size. So if an mRNA is associated to many ribosomes, goes to the bottom of the gradient, it sediments the bottom, but mRNAs that are associated with less ribosomes, they're more to the top. So when you measure the absorbance of RNA at 254, you get this very characteristic profiles, which are called polysome profiles. You can actually see the different peaks of the ribosomes, and you can see light polysomes, so 1, 2, and 3 ribosomes associated, and then heavy polysomes, 5 and more, where mRNAs that reside in this area of the gradient, they're heavily translated. We did not see any major difference in the polysome to monosome ratio, suggesting that global translation is not affected in this model. But when we looked at the polysome profiles and compared heavy to light fractions, we selected the 3 top genes from the data set that is the intersection between the shock and then the memory genes, as we would say here, right? And then the true memory genes, the genes that are not affected by the shock. And as you can see, the genes that are in this category, which is the same between the shock alone and the context plus shock, they are activated in both cases. Their translation shifts to the heavy polysome, so they're translated more in both cases. However, the genes that we predicted to be in the pure, let's say, memory gene subset, these are only activated when the context is paired to the shock. And this at the level of translation. More interestingly, to the broader field of neuroscience, when you look at FOS, ARC, and EGR2, some immediate early genes that I showed you earlier to be used to report neuronal activity, they do get activated both when there's a behavior that leads to memory, but also when there's just a shock, when the animal is shocked in the box, which does not elicit the behavioral response. So this was not the case, however, for the 3 targets, our 3 top targets from the transcriptomics. And as we predicted here, these 3 genes, they have a very clear response only when the animal learns the condition, the fear conditioned behavior in the box. So basically, the study at the time we were very excited. It did not get the attention or the the publicity that it should maybe, but we were very happy at the time that we were the first to show that the shock alone elicits gene expression, which are clearly distinct from the pairing of the conditioned stimulus with the unconditioned stimulus, which elicits behavior. So that was one of the first reports that there's specific gene expression to memory, which is not simply activated if you stimulate an animal. And this was one of the first studies that studied the translatome using ribosome profiling, and we did find that the AF2 alpha pathway is similar. Then Jonathan Weissman's group at the same time had a bioarchive that they published something similar, and they came also to confirm that the AF2 alpha is fundamental for long-term memory. So maybe one take-home message is that neuronal activity reporters such as FOSS, ARC, when studying behavior memory should be accompanied by appropriate controls, because they're not only activated by behavior or memory related stimuli, but they could be also activated by stress or by a shock, let's say, for example, here. So in the remaining time, I'm going to talk about the second project, which is linked to neurodegeneration. And this is the PhD work done by two brilliant PhD students in my lab here at Forth. Our approach here was more by the models and then moving on to our central hypothesis in the lab, which is protein synthesis. And at Forth in Ioannina, for the past 5 years, we've been working very hard to establish a facility which can derive pluripotent stem cells from patients, can use them to differentiate them to different cell types, and more recently to 3D organoids. We can do different types of analyses here. And recently, with a grant from the Hellenic Foundation for Research and Innovation, we'll be getting some new equipment to do physiology apart from the basic analysis that we will be doing so far. And having these models, these two students were very much interested to study Parkinson's disease and to develop models to study the disease. And I always had an interest to look at how protein synthesis is regulated in Parkinson's disease. So they've been working for me now for many years, and we've been able to develop models that we can reproducibly have either dopaminergic neurons as a 2D model, and then midbrain organoids, which is the area that is very rich in tyrosine hydroxylate, positive dopaminergic neurons. These are the neurons that die in Parkinson's disease. And having these models, we saw in the literature while we were working on this project that the Dawson group from Hopkins, they reported that in culture neurons and in animal models of Parkinson's disease, that synuclein actually is a player in the pathways that our lab is working on. And one of these pathways is the PI3 kinase AKT-mTORC1 pathway. Actually, what they very nicely showed in the paper, which they treated neurons and animals with alpha-synuclein fibrils, and it did show that synuclein physically binds to many components of the mTOR complex, one of them being TSC2, and thus it cancels out the negative function of TSC, which is to inhibit mTOR, and thus mTOR is hyperactivated. So it's a really nice study, and this also further spiked our interest to see how is protein synthesis regulated in this model. And since we were working on the models anyway, and we had obtained the synuclein lines from Tom Durkin's lab at McGill, at MNI at McGill University, we started trying to learn more about this new field for our lab, which is that of synucleinopathies and Parkinson's disease. And as the audience may know, because this is your broader field, native synuclein misfolds, and through oligomeric state, it forms this fibrillar alpha-synuclein, which at later stages of disease leads to Lewy bodies, these inclusions, which are seen in postmortem brains of Parkinson's disease patients. The status of alpha-synuclein clearly correlates with the status of Parkinson's disease, so aggregation and spreading, neurodegeneration, and the different symptoms seen in the patients. And it is now established that similarly to Alzheimer's, synuclein in this context is has a prion-like cellular spread. And this is what in areas of the midbrain, such as the substantia nigra, leads to degeneration, which underlies the motor and cognitive symptoms seen by the patients. And in these patients, apart from sporadic forms where synuclein is also a key player, there are mutations involving alpha-synuclein, duplications and triplications, either homozygous or heterozygous. And this is the model that we got the iPSCs from and used an isogenic line where the triplication was corrected. And what we did is two groups before us, the Durkin group and the Patikas, they showed that growing midbrain organoids with having the triplication of synuclein, they do develop cell death after day 100. So we wanted to see and study these prodromic stages up to day 100, where visibly the cells look healthy, even though they have some differences that I'll go talk about in a second. But there's no significant reduction in the number of tyrosine hydroxylase positive cells up to day 100. So we wanted to look at the early stages and see how protein synthesis is a key player there. So we chemically induced the midbrain fate in these organoids, and we developed organoids up to day 100. We do see that at day 100, the mutant organoids that harbor the triplication of synuclein are smaller. And this is also in terms of projected area and diameter. We do see increased expression of synuclein as expected by the triplication, and this synuclein, as you're going to see in the next slide, is also phosphorylated, but we also saw those neuromelanin depositions in the isogenic line where the mutation was corrected with CRISPR, and in the triplication line, it was much more prominent. As promised, we did see increased phosphorylation of alpha-synuclein already from day 50, which was more pronounced than day 100, but we did not see any difference in the abundance in the number of Th-positive cells at day 50 and day 100, suggesting that we're studying a part of the development of the model where there's no pronounced cell death or neurodegeneration per se, if that can be told for this type of model. So the first thing we did for this project is we ran a proteomics experiment with the organoids that were aged at day 100 to see if there's a change in the proteomic landscape. And indeed, we did see very pervasive changes in protein expression with very strict criteria. 79 proteins were downregulated in the triplication, and 187 were upregulated. The main pathways that we saw affected, represented in the gene ontology, this is a metascape analysis of networks. We saw presynaptic mechanisms such as dopamine, serotonin, and neurotransmitter release. We saw changes in cell adhesion in the Hippo pathway, but we also saw changes in axon guidance on L1CAM. On the other hand, in proteins whose expression was reduced in these models, we saw mainly changes in the extracellular matrix and some other motor and actin-myosin complexes. To further study the gene expression pathway, we also performed RNA sequencing analyses in this model. Again, here we saw changes which are actually very well correlated with the proteomics changes. So again, we see the same pathways. We see the synaptic pathways and the upregulated, basically, and then the ECM pathways being downregulated with a underlined here that we saw upregulation of perineuronal nets and the triplication, but overall ECM, we saw a decrease in the overall ECM in the model. And to answer the questions that really interest us as a lab, which is translation, we performed ribosome profiling and these midbrain organoids. This time we used a newer version of ribosome profiling, which uses an analog, which is a puromycin-like contraption, this 3P molecule, which all it does is, if it's coupled to magnetic beads, it isolates ribosomes which have a conformation that is actively translating, so an elongating ribosome. And by immunoprecipitating these ribosomes with the 3P beads, we're able to sequence them to generate libraries, extract the footprints, and measure global translation in this model. We saw changes which were not that pervasive. There were some very, very small changes actually in global translation, but we noticed that there was a large subset of genes which had an opposite change between transcription and translation. So while transcription would predict that the gene would be decreased, translation showed that this thing should go up. And we noticed that for genes that were upregulated, for genes that were upregulated in ribosome profiling, but were downregulated in RNA-seq proteomics, this concerned mainly synaptic genes and genes that have to do with the synapse, so the presynaptic role, let's say, of alpha-synuclein. But for the subset of genes that were upregulated in transcription and proteomics, but were downregulated in ribosome profiling and translational efficiency, we saw a correlation with gene ontology categories, enrichment of gene ontology categories that have to do with the extracellular matrix, mainly of the cells. And we wanted to confirm those. So as you can see here in the heat map, these are the statistically significant top genes that we believe the mechanism that's happening is called translational buffering. So as you can see, for example, here the top gene is aggrecan, which is a constituent of perineuronal nets and of the extracellular matrix. It's significantly downregulated in RNA-seq, but as it is in proteomics, but its ribosome profiling and its translation efficiency is exactly the opposite. And this is a phenomenon called translational buffering to prove that indeed translation is upregulated for some mRNAs that go down, or translation is downregulated for some mRNAs that go up in transcription. We again ran polysome profiles. We isolated the fractions. Again, we don't see a change in global translation between the polysome profiles, only slight between the triplication and the isogenic. But when we measured with qPCR the abundance of these RNAs in the different fractions, you can see in gray in the isogenic that this protein called TPPP is associated with heavy fractions, but in the mutant it shifts towards lighter fractions, suggesting that there might be a mechanism at play that tries to reduce the translation of this mRNA because the protein is going up. The exact opposite phenomenon was seen for aggrecan, which was the ECM constituent. Normally, it's very mildly translated. It's associated with light ribosomes here in the gray curve, but in the synuclein triplication line, it shifts towards heavier fractions. It's associated with heavier porosomes. It's a response of the cell trying to translate more of the aggrecan because its protein abundance is reduced in the cell. None of this was seen for a control gene such as actin. And because this pathway points towards proteostasis and the integrated stress response, we looked at the F2 alpha. And indeed, at early time points, there was no change in the phosphorylation of F2 alpha, but at day 100, there was a pronounced increase. But we did not see a change in general protein synthesis. And altogether, this suggests that around day 100 of this model, there is activation of the ISR, which could, to a large extent, explain the phenotypes that we've seen with the translational buffering and the discrepancy between ribosome profiling, RNA-seq, and proteomics. So seeing that ECM is a key phenotype and a key gene category which is modulated in our system, we wanted to study ECM, and we collaborated with Alexander Tichetev's lab in Germany. They're experts in imaging the ECM and quantifying and modulating it. Even though it seems natural, there's very little literature looking at the ECM in organoids. There's only a handful of papers that you can see stainings. So we had trouble establishing these methodologies, but it's clear that apart from the matrigel, which is one might contain some ECM components, the organoids themselves at ages that we see in this model, they do develop an extensive and complex ECM, and they do develop structures called perineuronal nets. So we stained with a stain that stains the perineuronal nets called WFA, and a protein of the ECM called aggrecan. So as we saw that the organoids develop extensive ECM, we designed a methodology to measure the abundance of this ECM, firstly in the pericellular environment, which is called the perineuronal net, and then in the area that's between cells, so the interstitial ECM. So we measured the abundance of WFA. And we saw something interesting. We actually saw a cell type specific effect of how the ECM was distributed in this model. First, at day 50, we measured MAP2 positive cells. So this is a marker for all neurons. And we measured TH positive cells. So these would correspond to dopaminergic neurons. Already from day 50, we saw only in TH neurons, but not in MAP2 neurons, a pronounced increase in the pericellular, so the PNNs, the perineuronal nets, and also in the interstitial ECM of the preparation. This was absent in MAP2, so it was not that all cells had elevated ECM in this preparation, it was just the TH cells. And by day 100, this phenomenon had also established in the MAP2 cells, and it still persisted in the TH cells in our organoids, as you can see here with the WFA stain for this lectin that WFA stains for the ECM. So together, while looking to study translation, we were able to replicate a lot of the findings that the Dawson lab showed about mTOR and hyperactivation of translation. In this 3D model, though, we don't see the exact same phenotype. We see a phenotype that points more towards a homeostatic activation of the stress pathway of the cells called the integrated stress response. And we do see that with the activation of EF2 alpha, but also with the very strong evidence from our multi-omics that in ribosome profiling, we see translational buffering. So the cell cannot cope with the protein load by day 100, and it's trying to buffer it by increasing or preferentially decreasing translation for mRNAs that have been boosted as a result of the accumulation and phosphorylation of synuclein in this model. So as the model progresses, and in the ages that we studied, which are before pronounced loss of TH neurons is seen in this model, we do think that the increase in alpha-synuclein by day 100 does change transcription with an unknown mechanism. We don't know how that might be. We see that this has an effect on translation, and most likely this is a clear phenomenon of translational buffering, and we do see altered protein abundance. Now, how synuclein might regulate translation, that's been shown by the Dawson lab. It could be by directly binding to TSC2 and regulating mTOR. And yes, we've also confirmed that these pathways are hyperactivated, but the the new thing with our results is that it seems that this mechanism might have potentially a relevance about the development of the disease as the ECM, the perineural nets, they form earlier preferentially around the TH neurons, but not the MAPT neurons, and this persists in ages before degeneration. And while completing the study, we went back to write the paper, and we did see actually that there is evidence in the literature that maybe the gene expression pathways that we saw affecting the ECM are indeed hyperactivated, or they do dysfunction, but nobody indeed has shown any specific changes or a significance as to why this change might happen in the ECM. So there is, however, a potential here, and there is, and we have a very strong interest as well to look if this could be a potentially converging mechanism for different forms of sporadic and familial Parkinson's disease. So this is something we're actively looking into. And with that, I think I've reached the end of my talk. I hope I didn't go way over my time, which I think I did. But I've been able to thank the people. These are the agencies that have funded our research. You can read more about the lab in our study, and I'm happy to talk to you more about our work or any potential collaborations we can have within HIAAD. Thank you very much.
Thank you very much, Christo, for this great presentation and very interesting data that you shared with us from published and unpublished work. Very exciting findings. We are happy to take questions from the audience. I will start with a question, if I may. You mentioned in the beginning of your presentation that the localized expression of proteins may affect neuronal function and may play a very important role, especially in neurons, since they are very elongated, and localized translation must be very meaningful for their function. So in this model that you are studying, Parkinson's disease, and for the proteins that you see in higher expression or lower expression, do you see that this might be localized? Have you looked at it, whether it happens in the synaptic sites or in the body of the neurons? Have you looked at it?
Yeah, so the short answer is no. We haven't looked into it, but there are a few good reasons, and I think this would tell a lot to the people about the model and their value for the disease and the community. Obviously, we're starting with iPS cells. The genetic clock has been reset, and this is a model where cells develop from the beginning. They auto-assemble. They do resemble a lot of the features of the midbrain. If you saw one of the SEM pictures that I showed, they tend to fold like in a very similar way as the midbrain does. Even the shape is dictated from early development. But in our case, and in the models that we showed in the other published versions, they do see TH cell death, but the clock, the biological clock of the organism, is really early. So it's early development, I would say, even at 100 days. Even though, I mean, the clocks are not aligned, and you cannot really say if day 100 of culture is day 100 in utero, for example. Still, you do see a lot of early markers being expressed. Having said that, we have stained with markers for mature synapses, such as presynaptic markers and postsynaptic markers. And we do see in our proteomics also that they are expressed. So yes, there are synapses. So the model has the potential to do more focused experiments, but you would look at local protein synthesis using reporters or using some other form of imaging. However, we've got to take this with a grain of salt, because the proper way would be to find a way to age the cells and have a model that better corresponds what happens in Parkinson's disease, where the patients, they tend to manifest the disease over the age of 50 or 60, unless it's a genetic form which manifests earlier on. So with those limitations, I think it's really important. Now, if I were to compare it to, let's say, cellular models like dopaminergic neurons, one of the things that we're excited about is that 2D cultures of neurons, they don't have perineuronal nets. This is a known problem of cultures of neurons. And it seems that the 3D structure of the organoids brings the model closer to what happens in the human brain, because we see the perineuronal nets. Now, whether this is a feature that persists into adulthood and not an early developmental feature, this is a very interesting question, in my opinion. But what I didn't say is that perineuronal nets have a protective role for cells in other parts of the brain. What happens in the striatum, what happens in the midbrain, what happens in dopaminergic neurons, this is still, there's only probably, I know of only one lab that has studied what's been happening in this brain region. So with acknowledging the confounds, some of the confounds of the model, which we really need to be aware when we study brain organoids, I think the fact that there are some strong phenotypes, interpreting them in the correct context would be very valuable for the Parkinson's community.
Thank you. Other questions?
Christos, can I?
Yes, please, Vaso, go on.
Thank you. Thank you very much. That was a great talk. Actually, we are dealing, because we are working with Alzheimer's disease and organoids, we are more or less dealing with the same questions, same issues about iPS cells and aging, and if you can see pathology and everything. So I was wondering how we can implement your findings, especially with the memory genes for Alzheimer's disease organoids. That's something.
Yeah, so I think there are two questions in the questions that I like them both because I think I can answer them. So the first one, this study that we did back in 2021, when looking at neuronal activation, I think we have three genes that I would predict would perform a lot better than CFOS and ARC. So, for example, if you had, let's say you did calcium imaging and then you wanted to make a reporter system in your iPSCs to look how specific neurons react to activity, I would say, why not try and look for a promoter for one of the genes that we published back in 2021 and not CFOS? That's one. The second part of your question was more about the organoids and
it was mostly about this very interesting upregulation of genes of specific long-term memory, and how this we can study in Alzheimer's disease models.
Yeah, I think in the organoids, the same problem applies to the question that Savas asked, is how mature are the synaptic circuits? Yes. I'm pretty sure you have electrophysiological phenotypes and you can see things with MEAs, and probably as your organoids age, they mature. I would say, I don't know, but definitely I would say these would be key players in synaptic function. And if you, for example, could boost their expression if they're low, if you see in your models that are low, they could act as a potential way to intervene and improve memory in Alzheimer's and other dementias, right? Where you would see, you would expect that memory genes, let's say I term them memory genes, right? But yes, they actually are, because we took out the non-specific control, which is the shock. So these are actually purely activated by the context, and this is the essence of memory that we think having a genetic basis for it, because mice cannot speak, they cannot tell us whether they remember or not. We need to correlate it to something. We can see how cells are activated, but really dissecting them, I would say that these genes have the potential to be used for treatment, if necessary, but through, let's say, genetics intervention, upregulating or downregulating them depending on the context.
Maybe you can use some of the Alzheimer's disease IPS lines that we have.
Yeah.
So we can look into this. Another question, maybe I didn't see correctly. If you go back to the slide about the ECM, I saw there was a big circle, and that was the cytoplasm of the cell. That was a little bit strange for me.
Yes,
Or
let
I
me
didn't
share.
see correctly. One of the last.
It's
One of the last.
how we quantified it, yeah?
Yes.
This one here.
Yes.
This one.
Yes.
Yeah.
So this
Yeah,
thing that you are circling is the cytoplasm of the cell?
No. So the black is the cytoplasm. Yes. Yeah. So the cytoplasm is there's no marker. We're just showing the WFA channel here. And we're just showing it because this is how we measured the thickness of the perineural nets and of the interstitial ECM, basically. So we draw this circle, then +1 micrometer, +4 micrometers. And then you measure each one of them. So when you get this measurement here, which shows pericellular ECM, that will be light green. I'm sorry, I apologize for the colors if someone could not see them, but yeah, light green would be the pericellular, and the interstitial would be the other one, the dark green, more towards the outside. So it's black just because we're showing one channel. This should be part blue. We have also MAP2 staining that I'm not showing you. So if you look at it here, actually you see the MAP2 staining, right? So this is the cytoplasm.
Okay.
Here, right? So the teal here is MAP2.
Okay.
Thank you. So we continue with the order. I see the raised hands in the screen, Era, and then Kostas.
Thank you very much for this nice talk, nice motto, and very nice imaging as well. So I have a question that maybe it's kind of a guess question. So you have an amazing data set of omics approaches and from your organoids, and you hypothesize okay, we don't know how from synuclein dysregulation we go to transcriptional remodeling and it's probably like the cause of this, let's say that the neuron is trying to do this translational buffering. Do you have any idea, because you have the data set from the mRNA components, whether there is really a problem at the transcription level and what that could be? And whether also it's deep enough, the sequencing to allow you to make this analysis and maybe find something interesting there.
Yeah, that points to a very big problem that we have with this study. So we tried to reduce the complexity by going to ages before day 100, right? So cells are not dying. That's one thing taken out of the equation. Then looking at day 100, for example, obviously, we didn't have the money to run omics every 2 days. So we chose day 100. We made a hypothesis. What do we see there? And then we tried to interpret the rest of the model based on that. So the status of day 100 is that there's buffering. We show that in the polysomes. We see this with ribosome profiling. So it means that the cell is trying to compensate for something by shifting mRNAs into or out of polysomes, right? Protein expression is up and down, so there's protein remodeling, and there is a change in transcription. Now, transcriptomic data, we know that for sure. They correlate very well. They have a R-square of more than 0.90-something%. I don't recall the number per se, but it's a very high correlation between the transcriptomics and the proteomics data.
Okay.
The correlation with the ribosome profiling is very poor because there's buffering happening. So stuff that's going up is going down. So it's exactly opposite. It's actually nearing almost an inverse correlation, which points to translational buffer. Now, the gene expression that we've seen, I'm a little bit hesitant to make a conclusion because my sort of problem with the model is that this is a developmental problem. There is a sphere that is growing, right, through autoassembly of cells. A lot of transcription is going to be required to increase size. A lot of transcription is going to be relevant to synuclein. And adding to that the problem that we don't know if transcription is the instigator or the result after ISR has peaked at day 100. So EF2 alpha is phosphorylated, the integrated stress response is activated. Does it also work with transcription? Or was transcription the instigator? It's a bit of a chicken-egg question, but we really don't have the capacity to dissect that now unless we do very acute experiments. And the Dawson study that used the fibrils showed that these pathways are actually acutely activated. So they add the fibrils, they see an effect, right? So there is hope that maybe this is something that is acute and we can actually study it. However, I'm afraid I think we need to think of this in a totally different way. Now, to tell you about the data set, we don't have the expertise, so we haven't looked into transcription regulation. We're a translation and control lab, so we do know we've looked at our UTRs, and we've looked at our 3' UTRs and translational motifs, but we don't have the capacity, and we haven't looked into transcriptional mechanisms. But the data set is there, and I guess we could look and see if there's, like, say, an upstream regulator. There are some gene ontology tools, I will give you that, but I've never found myself something meaningful there, which is stands out or makes sense. So to summarize, nothing obvious, but I think there are many caveats to how the question should be answered. So yeah,
which maybe can be corrected for all the variables and just focus on the triplication effect somehow with also other data.
Yes,
because I know that there are also other triplication, and I know that you got the line from Edinburgh, if I'm correct. Yes.
It's actually, so it is the
it's the McGill. It's the McGill line.
We got it from McGill. It is the original line is from Edinburgh, from Tilo Kunath. It's Tilo Kunath's lab, but the original Tilo Kunath was not there was an issue with the isogenic control. So this is the Durkin isogenic control, which is corrected at McGill. But the patient line is from Tilo Kunath. Yeah.
Okay. Thanks.
Kostas, please.
Thank you. Really nice talk. I'm sorry, Vasa had to go because my question is relevant to her work. So as a translational epidemiologist, I'm always concerned about inter-individual variability. So for example, Vasa's lab has shown in organoid models across different people, that across people, there's variability in a number of these parameters that you measure, and especially in drug response. So my question to you is, how many different people contributed to the cell lines, and how would the results you think look if you had, say, and I know this is very expensive, let's say you compared across 10 different people, 5 different people, to get a feel for the variability in the specifics, what's variable, if any?
Yeah, I don't have any data to answer your question, but I can elaborate more on the experimental design, because what you just summarized very nicely in a sentence is one of the biggest questions in the field of organoids, like how can we trust data that come out of all these studies that show differences? Will they be reproducible? And as you said, expanded to the population, taking into account the variability within the population. So the recent papers in organoids, they have all these standards which are very similar to the International Society for Stem Cells, as having more than one lines, having batches. So we did all that. We have different batches of organoids. What I didn't show today is that we also have data on a knockout line. And this is very interesting. So not us, Tom Durkin's group, they took the patient leg and they removed all copies of synuclein. And we do see phenotypes there as well. And these are in I didn't have time to present it. They're in the preprint that we have, and they'll be hopefully published soon. We're under revision at the moment. The fact that the knockout affects the same pathways, but in a different way, is very encouraging, right? So batches, replicates, knockout, this is the only support we could have for the design. Now, regarding your question, we haven't tested in individuals. What I would predict, because several labs, so Struder and other labs, have reported that other mutations like Parkin, PINK, and GBA, they have changes in the gene expression. I think the changes in the transcription of the ECM is a feature seen across many genetic forms of Parkinson's, maybe in some sporadic forms. That is a given. So I would predict then, if that is the case, that if we had organoids from different individuals harboring, let's say, just the triplication of synuclein that we studied, I would say at least the change in transcription would be replicated, even though there is variability, because I think it's a core phenotype speaking to what synuclein does. We see it in the knockout also. So knockout has an effect on the ECM. So it's not just the triplication. So it seems to be correlated to a very central function of synuclein, either through mTOR, as the Dawson lab showed, so by doing it through mTOR, and mTOR has a link to the ECM, or perhaps through an entirely new pathway. To me, I find it fascinating because the prion biology of synuclein has a very close intersection with perineuronal nets in the ECM. So apart from the cell autonomous role of synuclein, it's really very interesting to look at what happens when synuclein exits the cell and it encounters different perineuronal nets around TH neurons and MAP2 neurons. And this is something we're trying to do now as a follow-up to this study. So do they act as nucleation sites? Do they act as protective nets for the TH neurons? Jessica Kwok's lab has done some work on this, and she's shown a few things about perineuronal nets in that context. But other than that, it's a pretty virgin field. Sorry, I went a little bit into DCM because that excites me, but I hope I answered your original question about the variability in the population.
You did, and I think your starting point was the best answer. There's no data. There are a lot of predictions. So I hope that labs increasingly demonstrate that because when it comes to drug testing and the generalizability of drug testing models, because there is such demonstrated variability, how are we going to decide which humans we give these drugs to?
Yeah, exactly. So yeah, I'm afraid I don't have a good answer there, but yeah, the approach that you're taking with trying different individuals or even engineering lines of different backgrounds, that's the way to do it. And yeah, we have the capacity to do that now. We didn't have it 5 years ago when we started this, so I think that should be one of the priorities to ensure that the findings are translated better to the clinic.
Great work.
Thank you.
There are no other questions from the audience as far as I can see. So thank you very much, Christos, for the great presentation, stimulating and raising many questions and giving back a very interesting discussion between you and the people attending the meeting. Thank you, everybody, for following this talk today. We meet with each other next time in the next presentation. Bye, everybody.
Thank you very much for having me.
Thank you, Christos,
Bye.
very much.
Have a good day. Goodbye.
Bye.